In September of 2025, I had one of the most important interviews with Christie Grace. She is a scientist who specifically, I thought, identified one of the biggest unknown risks when people are talking about the safety of mRNA technology. It was so important, she published a paper on it. I had the interview. She wanted specific research to be done about adducts, and this has largely gone unsaid or uninvestigated. How I knew I was over the target is that when I spoke about this, I was being censored left, right, and center. And so I knew this was really important science. It came back today because I saw a recent post about discussions in Germany in some kind of coronavirus inquiry. I'll clarify. I think there is a formal inquiry and there is a parallel inquiry that is organized, I guess, back from an alternative sector. The point, though, is that what was being discussed there was extremely relevant. I'm going to play some clips for you, and I'm going to then explain some of the basics about what are adducts and why this question is something that nobody wants to focus on because the impact is so absolutely significant. So let's just quickly go and listen to what was said. I got this from Dr. Asim Malhotra on his YouTube website, to give credit for that. And listen, it's in German, but there's a translation, and I will jump in from time to time to give some clarifications. So let's start off with what was said here.
Herr Sterz, Sie haben als Cheftoxikologe für Pfizer Europa gearbeitet. Ist das korrekt?
Ja, das ist korrekt. Ich war dort verantwortlich für alle Tierexperimente, die der Arzneimittelsicherheit dienen.
Danke. Meine Fragen beziehen sich auf Comirnaty von Pfizer-BioNTech. Sie haben sich intensiv mit der Zulassung dieses Impfstoffs befasst, mit den Unterlagen. Ist das korrekt?
Das ist korrekt.
Wurde die Karzinogenität dieses Impfstoffs vor der Zulassung überprüft?
Nein, das karzinogene Risiko wurde aus Zeitgründen nicht untersucht. Ich finde es übrigens sehr bedenklich und auch bedauerlich, dass keine alternativen Untersuchungen durchgeführt wurden.
Just pausing that here a minute. What he's saying there, because when you're using any kind of technology, especially a novel technology at scale, you want to look at two things: carcinogenic potential as well as genotoxic impacts. And because of time, what he was saying, they were unable to do this because they needed to get the vaccine out as quickly as they could. I think that's a reasonable thing to say. However, what was promised on the documents was that parallel, ongoing animal studies would be done to look at some of the risks that couldn't specifically have been clarified very early on because there was a time constraint. That was the starting point. Let's listen some more to what he had to say.
Wir beobachten in Deutschland, aber auch in vielen anderen Ländern, dass die Geburtenrate nach der Impfkampagne einbrachen.
Sie spielen auf Untersuchungen betreffend die Vermehrung an. Eine Studie an Ratten wurde mit Comirnaty mangelhaft durchgeführt. Damit waren keine verlässlichen Abschätzungen zur Wirkung des Impfstoffs auf die Trächtigkeit oder die Entwicklung der Nachkommen möglich. Man hatte aus der Contergan-Katastrophe nichts gelernt. Die Zulassung erfolgte laut RKI-Protokoll im Schnelldurchgang. Das bedeutete, dass essentielle Toxizitätsstudien der Schnelligkeit geopfert wurden, ohne akzeptable Begründungen. Ich kenne bei vergleichbarer Indikation keinen Fall, in dem all diese Studien weggelassen wurden. So führte die Zulassung zu verbotenen Menschenversuchen.
Pausing that there again. So what he's saying is that he's never seen this done previously where these kinds of studies were not actually done. And he's right, because this was very important. But once the safe and effective narrative came out, I suspect that regulators were no longer pushing. And in business, which is what pharma is, why would they go looking for problems if they are not being specifically asked by regulators? You may argue that they should try and do it anyway, but it would be cost and risk to them. So in a sense, the question does come back to regulators. Why were regulators in countries across the world who are supposed to be independent, why did none of them ask for these bits of research to be completed for them to have clarity on what is happening? Now, this is the bit, before I continue with what he was saying, I'll just give you a few points about the issue that was raised by Christie Grace and adduction. So a lot of people are talking about the mRNA itself making the spike protein and the risks of genotoxicity or carcinogenic impacts with regards to impurities. Those are all real and good questions. But there is something that is so big that nobody wants to touch it, and very few people understand it. I had never heard about it prior to Christie's presentation. And as I said, the link to the full interview is in the description below. It is mind-blowing science. So here is just an insight into what she had said. When we're talking about the vaccine, the COVID mRNA vaccines, they are put in these cationic lipid nanoparticles. They have on the outside certain phospholipids, and inside they have the mRNA. And this is so that it can get into the cell without the immune system destroying it. That's the whole point of these pieces of technology. And this has to be protected in order for the body to be able to process it. So once the body or the cell, it should be a muscle cell, gets the mRNA, after it has then entered the cell with the lipid nanoparticle, it then produces spike protein, which triggers the immune response. And that's the principle. Now, here is the bit that very few people take into consideration. The lipid nanoparticle itself is very complex. It is not a simple thing. It is actually paralleling or replicating something the body does in terms of exosomes. So it produces these packages that can be shared between cells. And this is kind of what it looks like. I got this from Therapeutics. And what you have is these are all the pieces: cholesterol, ionizable lipid, PEG lipid, phospholipid, and the RNA strand. And all of this is packaged together. Look very carefully, and you'll notice that the mRNA here is surrounded by these ionizable lipids. And it's held in place and safely kept there so that when it gets inside the cell, this is not damaged and can produce spike protein. The question becomes, once this has delivered the mRNA, what happens to the ionizable lipids? How does the body deal with it? And what risks does this carry? This is the part of the question that hasn't really been addressed. And it's one of the very big questions that is a huge hole in the science. Very few people know about it or even understand it, but Christie Grace did. And she absolutely dissected exactly what this was and why it was relatively easy to prove. And this point has been missed year on year, month on month, because this was actually so big that when I spoke about it before, she said this was so serious it could take down the whole platform. But nobody seems to know, and nobody seems to still be interested. But let's listen some more to a few more minutes of this video here.
Also das RKI hat damals ja intern notiert, dass die Nebenwirkungen und Impfschäden erst nach der Markteinführung geprüft werden sollten. Was ist dabei herausgekommen?
Und Pfizers Postmarketing-Bericht sprach von über 1200 Todesverdachtsfällen innerhalb von nur 2 Monaten nach der Zulassung. Spätestens da hätte man Comirnaty wieder vom Markt nehmen müssen. Das Paul-Ehrlich-Institut erhielt, wenn ich richtig informiert bin, bisher 2133 Meldungen über Todesfälle nach Comirnaty. Bei diesen Spontanmeldungen gibt es eine hohe Dunkelziffer durch Underreporting. Die wirkliche Anzahl liegt also viel höher. In den USA geht man davon aus, von einem Underreporting-Faktor von 30, mit dem die registrierten Fälle zu multiplizieren wären. Für Deutschland entspräche das 60.000 Todesfällen durch die Impfung.
60.
I'm pausing that here again. There's an important point that I got from another interview with Hedley Rees inside Pharma. And he highlighted a very important point, which is that the batches that were used initially were kind of like the test batches. And in order to scale up, you would tend to find that you had more complications with the initial batches than with subsequent batches. And this is why some people find that you can mirror the side effects almost to specific batches that were done. And when we think about that, what it probably means is that the risks that were present early on with regards to the rollout of the vaccine may have somewhat been mitigated later on, which is why we didn't see the same kind of side effects with later dosing. And that's something to take into consideration when looking at some of the issues.
60.000. Also die Bundesregierung hält ja die wichtigen Safe-Vac- und KV-Daten zu Impfschäden zurück. Und die Mehrheit dieser Kommission hat es sogar abgelehnt, diese Daten anzufordern. Hätte man Impfschäden und Todesfälle bei einer regelkonformen Zulassung vermeiden können?
Ja, denn regelkonform hätte Comirnaty gar nicht zugelassen werden dürfen.
So derzeit kämpfen viele Impfgeschädigte in Deutschland um Schmerzensgeld und sie verlieren oft, weil die Gerichte sagen, Comirnaty habe ein positives Nutzen-Risiko-Verhältnis. Ist diese Annahme berechtigt?
So you can see where this is going. I'm just pausing this here because it's in German, but you can see the translation. The point being is that one of the other mistakes that has been made is that there has not been enough post-marketing pharmacovigilance looking at longer-term effects for many people. Very often they focused on 4 weeks or sometimes up to 6 weeks where people may have had adverse effects, not recognizing that they should also have been looking carefully at what were the effects when people got reinfected. So the reinfection adverse effects in terms of vaccinated cohorts should have been also something that people would want to look at. So there are many gaps, and even the recent ACIP, and this is from the USA, the Immunization Practice Group that was looking at vaccines, they wanted to bring in a specific coding, ICD coding, that would have been able to identify COVID vaccine injuries. This has probably been lost because that group has been disbanded, but it highlights that there are some very, very significant gaps in the science that, in truth, very few people are interested in looking at. And so here is the final bit I'll say with regards to Christie Grace's point. I'll just show you this here. When we look at the lipid nanoparticle, and as I said, with regards to the ionizable lipids, they will hold the mRNA in place and protect it from being degraded. So once this gets inside the cell and this is removed, the mRNA is free to then make spike protein. But now you've got these ionizable lipids inside the cell, okay? And so Christie Grace's question was, what happens to these ionizable lipids? And this is where she mentioned the point about adducts. And what happens is that if these ionizable lipids get a positive charge on them, they can then bind to other proteins inside the cell, and this can become a risk in terms of cancer. One of the best examples of this is Zantac, or ranitidine, which was removed from the market because of the adduction risk and the carcinogenic risk in the longer term for people who are using it. So this is well known in the pharmaceutical industry. And so this question was partially asked, meaning that they did start a study. They saw that at certain temperatures, the adduction levels increased, but it was never followed through. And so what she was saying was that this research was not expensive, could easily be done to clarify what happens with regards to adduction. And when I had done some work here, this is just showing you an ionizable lipid, and this is what happens if it's positively charged. So you have to just remember, not all of them. If you have 1,000 of them and just say 50 become positively charged, they can then bind to specific proteins. And in this example, I use hemoglobin. Once it binds, that positive charge on this protein alters the shape and the configuration of the protein. Similar example here was with fibrin. If this then binds to fibrin, you get an altered fibrin. And what I was pointing out was whether or not positively charged ionizable lipids could have been contributing to abnormal patterns with embalmer's clots. That's when I was looking at that. The main point is that these proteins no longer function in the same way and therefore could represent a risk to the cell and a risk overall to the body. Adduction formation. So my point is, if you don't know anything about this, the minimum you can do is get educated about this concept of adducts. It is a heavy interview. The link is in the description below. But if you understand the significance of it, you will realize why people focusing purely on mRNA has been relatively easy to deflect, because they will always argue about, well, show me the proof and so on. If you focus on adducts and you corner the scientific community to look at whether or not adduction is occurring, where these adducts are forming, and if they are forming, what is the risk, genotoxic risk, carcinogenic risk? Have you clarified it? There is nowhere to go. And this is why this point is such an important point. And that's where we need people to ask these kind of hard questions, because that question is very difficult to deflect, because any good scientist working in industry would know that that is a question that they should have an answer for, and they don't. And that was the whole purpose of Christie Grace's paper, was highlighting that gap and emphasizing that there was a responsibility on regulators and on industry to get clarification on that point before they continue to expand the concept as to what is being used. You need to know how the body deals with the particles. And just in case you are just joining, this is what we're talking about. Just a reminder, this is what a lipid nanoparticle is composed of. Her question was, these thousands or millions of ionizable lipids that are inside the lipid nanoparticle, once they have released the mRNA, how does the body deal with them? And if they become positively charged and bind to other proteins, does it have a genotoxic or carcinogenic risk to the individual? These are big questions, and these are the kind of questions that need to be asked. Thank you very much for joining me, and look out for more information in the near future as we continue to ask the hard questions.